Hello guys, welcome back to another video. Today we'll be looking at ecology, and today we'll be looking at ecology part 2. So the contents of ecology we'll be looking at today are adaptations, biodiversity and sampling. Plant adaptations. So the adaptation that plants have is to dig deep roots to find water, have shallow roots to absorb rainwater, and thick stem to store water. They also have leaves which are reduced to spine to surf especially in deserts, which is an example of this are cactuses, where the leaves are reduced to spines to reduce surface area for water loss by transpiration, because there's very limited water in deserts, so they have to have very small leaves. Also, spines also prevent animals from eating the plant to get water, because that's what plants normally do. And also, they have curved leaves to protect water loss from the stomata. And also, waxy cuticle on the leaf to prevent water loss. So the main theme here of plant adaptations is to prevent water loss. So the ways they do it are waxy cuticle, spines, curled leaves. Animal adaptations. Animal adaptations. Animals have thick fur to provide insulation and stopping heat escaping. A perfect example of this is a polar bear, which has really thick um, white fur, and the white reflects off sunlight, as opposed to black, which kind of absorbs sunlight. Also, animals have large paws to spread the weight of the animal and prevent it sinking into the snow. So the that means a polar bear have really large paws so that they can stand upright on the snow, even though it's quite deep. In addition, polar bears have small ears to reduce the SA to volume ratio. Small surface area means less surface for the heat energy to escape from, so they can contain all the limited heat energy they have, and so be able to survive in the extreme temperatures. Also, they have a coat that provides camouflage so predators or prey are not easily seen, and they can camouflage themselves in the snow really easily. Also, they have long legs, necks, or big ears, which increases surface area to volume ratio. This allows more heat to be lost to cool down. So when we talk about long legs, long necks, or big ears, what mainly an example of this is the elephant, because they have really big ears because they live in a really hot environment. And behaviors such as being nocturnal helps avoid predators, or because it's cooler at night. So that's what bats do. Abiotic and biotic factors. Abiotic factors are environmental factors that are non-living. On the other hand, biotic factors are environmental factors that are living. So abiotic factors can be chemical or physical, but not biological. So important abiotic factors are light, light intensity, water, the pH of soil, temperature, um, oxygen, and carbon dioxide. On the other hand, biotic factors are environmental factors that are living. So these are pathogens, competition for mates, competition for food, and food. Sampling. So now we're looking at rules for sampling a field or wood. So step one is to place a quadrat randomly within area to prevent bias. Step two is 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 splitting the quadrat into squares, each to see the variation of species, and it represents an overall percentage of the full square. Step 3. A quadrat with 25 square would mean each square was worth 4%. You could count a square if the plant takes up half of the square. Also, step 5, you add up the squares to tell you the estimated percentage cover for that species. Step 6, you collect multiple random sample readings to calculate a mean. So this is called quantitative sampling. So there are two types of sampling, which are So the two types of sampling are random sampling and systematic sampling. So quantitative sampling is another type of sampling. Also, a quadrat is a square frame used in biological sampling. Sampling is a process of recording small amount of information to make wider conclusions. So it's kind of like a microcosm. Random and systematic sampling. Random sampling uses a quadrat involving placing of quadrats at random coordinates. Regardless of where you're investigating the number of individual species, the species diversity, or the percentage cover in different areas, you would use random sampling. On the other hand, less commonly used systematic sampling can be used if there's a trend or pattern across the habitat. Also, another key thing that's used is a concept. It's a line along which a systematic sampling occurs. So the systematic sampling occurs only on that line. Also, one more definition you will need to know for this are what concordant results are. They are results that are similar. Required practical. So this is a required practical in the GCSE course. The required practical is measuring population size in a habitat. So the aim is to measure the species richness on the school field in areas which the grass is regularly and irregularly cut. So method. So choose a starting point on the school field in area where the grass is often cut. You use random numbers to generate a series of coordinates to place your first quadrat. So this is using random sampling. Then you count the number of different plant species within this quadrat. So how much of a species there is. Then you return to your starting position and repeat step 2 and 3 a further 14 times using different random numbers. So repeat step 1 to 4 for a part of a school field which your grass is infrequently cut. So you can compare results by calculating a mean for each location. You can see what place is more diverse, what place has the most species richness.